So, evolvability was a term I think first introduced by Richard Dawkins in 1989, but that concept has been around for quite a long time before that. But actually, it appears to describe many separate related concepts. I'll describe three sort of main ones that I've sort of distilled from this literature, which I'm no expert in whatsoever. So the amount of variation within a population is one measure of evolvability. So you see here are two kinds of snakes, actually the same kind. They're both king snakes. Actually, I used to catch them when I was growing up in San Diego. They're the same species, but there's a large variation in the patterns on the skin. And so a large variation would mean, by this definition, or high evolvability. Another sort of definition you could talk about would be something more to do with the mutation rate and how mutations propagate. So here I have two different crocodiles, regular one and albino one. And there's some mutations you need to go between these two different kinds of phenotypes. And you could think that how mutations propagate are going to affect the phenotype. And so this will be a measure also of evolvability. If you're getting a lot of mutation, a small number of mutations, but it makes a big difference to the way the critter looks, then you have a high evolvability by this definition or notion of evolvability, right? So that's another way you can think about this term. And finally, the most difficult one to understand, and maybe most related to this conference overall, is innovation. How a species sort of tunnels between one phenotype to another. So here we have, to me, maybe the most extreme example of this, and that's the formation of a rotary motor. So there's only a couple of examples of this sort of thing in biology. Bacteria possess them, and you have them in some forms of ATPase, where in fact somehow through evolution, maybe someone who knows more about this can give me a path, but you have a different topology when you go to a wheel. So this is actually a wheel, right? It rotates around, and these are collected to this filament, which rotates around and propels a bacteria around so it can search for things to eat. And so for this to have happened, you can see that it would be very hard to see how this would happen in a kind of continuous way, where you have a change in topology like this, and we don't have wheels, and it'd be very useful in lots of situations, and it's probably because it's not an easy thing to evolve. But as Daniel Fisher has pointed out, there have been about 10 to the 40 bacteria that have lived on Earth, which is a very large number. And we have numbers that big, you can start getting very unlikely things happen. And so this is an example of a kind of thing that is also going to be very important in understanding evolvability, is this sort of innovation that you can get. Okay. So now you can ask the question, is evolvability evolvable? Okay. So this is there's a paper by Massimo Pigliucci on this from 2008, which is very interesting. And I'll sort of summarize what he says and others say in this area. So yeah, so the idea is that your evolvability, can you actually evolve that to make you more evolvable or have more evolvability, right? Is there some way of doing that? I mean, the sort of way that we tend to think about evolution is this sort of dumb, and you just get random mutations, and wow, sometimes they work, and you get, intelligence or whatever it happens to be, or you can swim like a fish. But is there more to it than that? Is the actual process of evolution itself under evolution? Is evolvability evolvable? And one example of this is that the mutation and recombinant rate depend on the species you're talking about. There's a pretty big variation depending on the species. Even if they're not too different, you get pretty big changes in these two things. And even more startling than this is that there are quite a few examples of proeukaryotes that they can modify the rate that they mutate. They decide to do this in situations where they feel they're under enormous stress. So you can put a bunch of bacteria in some antibiotic, which is killing them off, right? You could be swallowing antibiotic pills or whatever, right? Or you could put it in a petri dish. And in the petri dish, when you do this, you can see that they actually have very sophisticated genetic apparatus to produce genetic changes. So if it thinks, oh dear, well, I'm in terrible shape here, I'm going to die, it just starts mutating like crazy. All these bacteria just go crazy and figure out a way of increasing the mutation rate in certain areas of the genome so that they can figure out a way to become antibiotic resistant. So there's certainly a lot of examples of this kind of thing. But how far can you take this? How evolvable is evolvability, right? How far does it go? And how do you even think about this sort of a question? Okay. So I'm going to switch gears now and talk about, as I said, something that seems to start with to be quite different. So artificial intelligence and neural networks. Again, I don't know anything about it, but I'll talk about it all the same. We heard from Bart Selman, who's an expert of this more, but I'll sort of summarize it as best I can. So intelligence, what is that? So the way I'll use it is essentially using knowledge to make decisions to further goals, right? That's essentially what you're trying to do. One of the major goals of artificial intelligence is to create systems that learn tasks, for example, like winning Go or doing facial recognition. And sometimes it can be quite hard to do facial recognition, is a decider right there. And here's this Go, which is, as you probably have heard, they're doing, they've got a machine now which is incredibly good now at playing Go, even though a year ago people thought it was essentially impossible, from what I understand. Now they're really good at it. So AI is changing, very rapidly now. And these are sort of things that also facial recognition, where they made huge strides recently, right? So the way that it works, though, is still different than the way we do it to a large extent. It's trained with many examples, right? You take many pictures of people, and you'll try and train your neural network based on many, many pictures of maybe the same person even, right? Okay. And the way they work, say, deep learning, which is sort of the same thing, as far as I can see, as more or less as sort of the 1980s sort of neural network community talked about, was to have these sort of hidden layers where you have these neurons sort of here at the inputs, and you're trying to learn a bunch of different rules. So you might have the rule 100110, and then you have some output you would like to learn, like 10, it's supervised learning. And you have then a bunch of connections between neurons in here, and the neurons can be simplified to be essentially sort of ones and zeros, if you like, or they could be continuous. And there are all these connections between them, right? And what you do when you learn is you try to figure out a way of modifying these connections so that when you put the ones and zeros in, that this sort of circuit will eventually produce a 1 and a 0 at the end. Okay, so that's the kind of thing that you typically do, but obviously there's a lot of complicated math that goes into really learning things like pattern recognition for faces and so forth. But one thing that all these things seem to have is high connectivity. Neural network models, as opposed to models of circuits that we use in chips, they have very many inputs. Just like the human brain, which might have thousands of inputs per neuron, or 10,000 inputs per neuron even, right? So the high connectivity, it does lots of important things which are important for the brain or for artificial intelligence. One is robustness. It will still work with some sort of pruning. You can prune these connections between these different neurons, and you'll see that you still get the same output most of the time, right? And that's very different than you would get for a normal digital circuit. You take some sort of CPU and you sort of start kind of connections, and pretty soon the thing won't work, right? But in this sort of an architecture, it's very robust to that kind of thing, which is good for the brain, right? Because you have a lot to drink and you still want to be able to understand things the next day, even though your brain has been a bit damaged. And another thing is that when you have a lot of these connections, it allows for the simulation of many examples. So you can learn many separate faces, if you like, right? You're learning many patterns, so you could put in many inputs and you'll get the correct output. So that's important just from an information point of view, because these connections contain information. So you can store essentially more information this way. It will be able to respond correctly to many separate inputs. I should mention, though, if you have too many connections, you don't generalize well. You become essentially less intelligent. You don't see simple, parsimonious ways of collapsing the data. And so neural networks still, as they're people use them. It's still a bit of an art to make them work well. Okay, gene regulation. So this is something that appears to be quite different again. So gene expression is something very important in your body. You have, we heard before, you have liver cells, you have brain cells, you have hair cells. You have the same DNA, but the way they're expressed in different cells is different. And it's controlled by a very complex mechanism involving other genes. Okay? And so you have these genes called regulatory proteins, or these proteins produced by these genes, that sometimes enhance and sometimes they repress transcription by binding to different points along the DNA. So what you really have is a circuit. It looks kind of like a digital circuit, from my point of view, at least. You have sort of inputs going into your into your regulatory network, and then you have some outputs. You would maybe want to produce more hair or something, or you might want it to be warmer or something. You want to change the outputs in some way, the output proteins that are produced. And so you have all these genes in the middle, and they're sort of regulatory elements that are upstream of each gene, and they sort of suppress or enhance each gene. So it really is a circuit. It's like a digital circuit that, the way I've drawn it here is quite sparse. So by changing the regulation, you can make this circuit do what you want. 111 might go to 10 or something. Okay. So gene regulation, I'm sort of thinking of it now in terms of digital circuitry, right? Now let me go on to something else now, non-coding RNA. So this was recently quite recently people sort of dismissed it as junk. Not entirely, but to a large extent. It's just this stuff that sits in the genome, and the genome is huge, and most of it's not actually used to create proteins. So people thought it didn't do very much. But on the other hand, it produces RNA molecules. Okay, so there's a lot of RNA molecules that don't actually go ahead and produce proteins. They just sit around. Most of what you're producing in terms of transcription, it just sits around in your cell, and it's not ever going to do anything like produce proteins. And so there's a project called the ENCODE project, which has got terabytes of information to figure out what this non-coding RNA actually does, right? And one thing that it does, certainly, is it regulates genes. In a myriad of ways, it's still not that well understood. There have been quite a few criticisms of ENCODE, this project. So the project claims that 80% of the genome has a biochemical function, but only 1.5 codes for genes. There's an argument about quite how much is functional. I'm not going to go into that in detail. It's sort of interesting to read about it. But one of the main criticisms of it is that non-coding RNA often seems to be just sort of randomly mutating at a high rate. If it was evolutionarily important, it wouldn't be mutating as fast as it is. So it doesn't seem to be an evolutionary constraint. So it's sort of neutral. It just sits there, mutates away, isn't doing any good. It's just wasting resources and producing a bunch of useless RNA. That's kind of the argument, because it has a high mutation rate. But on the other hand, I mean, the function of these is still unknown to a large extent, but a lot of them is still known. And it's definitely, a lot of it seems to regulate proteins. So the physics of it is like this. So I'm going to get into more biochemistry now. So RNA is quite sticky. You can go to databases online. You can type in random RNA sequences. They'll fold up. They got some charge to them, but they do tend to form secondary structure like I sort of have here on the right-hand side. And they're quite dense. They're quite close together. And so you're going to have some sort of a soup full of these RNA molecules, where some are bound to each other and some are free. And they're going to sort of have some sort of chemical equilibrium between them. So if you consider this sort of chemical equilibrium, we have these different RNA molecules. We have different couplings between them, different equilibrium constants, right? And you can solve this pretty simply to figure out what the amount of unbound RNA will be as a function of the equilibrium constants. And you get this self-consistent equation for this big soup of reacting this sort of equilibrium concentration of the non-coding RNA. Now, if you've done any machine learning stuff, you'll see this. This is pretty similar to, JIJSI. It looks a lot like what you get with machine learning algorithms. So the question is, can you make that analogy more precise? So here we start off with good old-fashioned cis regulation where you have sort of fairly sparse network. What happens if you were to add non-coding RNA? Then you have this big soup of RNA that's produced. It's not supposed to do anything, but wait a second, it's there. It's going to interfere with the RNA that you want, the messenger RNA. It's going to do something. What's it going to do? Well, it's what I said before. We can solve for what it's going to do, and it's going to bind to a lot of the RNAs. It's going to screw up how much RNA is produced. Biology doesn't like that. It's going to, at some point, become useful. All this extra RNA floating around, clearly it's going to have to still be useful to have. And so the question is, if I do a simulation of this, can I see that it's doing something interesting? So one thing is a fluctuating environment. Say I had an ice age going between hot and cold like this. I could ask, what happens if an organism has to change the rules that it's using depending on, like growing fur, or there's a new predator. There's some oscillation in the environment. So I have to change this learning rule I showed you before. So we can try to change it from something like this to something like this. And then we can try to train our network with and without non-coding RNA every, say, 1,000 generations to see what happens. And what you see is that as time goes on, so we switch every 1,000 generations, we switch from one rule to another. And you're making mistakes, and then you're making mistakes. But eventually, after a very large number of generations, you manage to adjust very quickly to the new condition with a non-coding RNA. That's because a non-coding RNA is storing the past. It's storing the ice age and all that information. So you can utilize that to very much more quickly mutate to the new environment. So if you think about it this way, you're really thinking about a problem which is very much like that of learning in neural networks. So here we have a distributed neural network, right, what I just talked about before. It learns from examples. And it can even be taught to evolve using the same kind of algorithms, evolutionary algorithms. People use those actually sometimes. Now we think about a genetic net with non-coding RNA. So RNA is highly distributed, just like over here, right? And it learns from its history, just as before you learn from examples. And it will evolve, right? In the same way that this network will evolve to become good at learning its tasks. So can a species be viewed as having some sort of intelligence? A species, not an individual, but a species. Here we have like a timeline, and you have a bunch of males and females of some species that are breeding with each other. And here we have in this blue background is supposed to be the environment, right? So as time goes on, the species are evolving to adapt to the environment, and the environment's changing in some correlated way. As time goes on, you expect that you'll be able to predict to some extent what the new environment is going to be. So you expect the genetic network to start doing predictions to learn the future so as to evolve the most efficiently, right? So basically, can it have intelligence? So because it utilizes historical information about the past environments to computationally predict the future, how to mutate, right? And you would expect a fluctuating environment would require greater intelligence. If the environment's constant, you don't expect to see, it's not a hard problem. But if the environment's challenging, it's changing all the time in a correlated way, then you're going to have to be smart on how you mutate. So it's really the same problem as in AI, where you see that the faster the gene network learns, in a sense, the fitter it's going to be, right? So that's the same as the real neural nets that learn concepts. And the more quickly they learn concepts, the smarter they are. Okay. So learning new concepts in AI is really like a gene network learning. Okay. But the thing is, actually, and that's one of the main problems we have today with AI, from what I understand, is that one of the biggest challenges is to learn quickly. You might have to take hundreds of thousands of examples to learn a new task when you have an AI network, right? You want to know how to speed that up. And there was some interesting work recently on learning how to recognize letters, which just use a very few number of examples. And that's the kind of thing that you expect is going to be necessary to do a better job with making AI more intelligent. So the same is going to have to be true with gene networks. Somehow they should be evolved to be intelligent. So we've really only begun. This whole thing really is not many people talking about this, but I think we've only begun to understand how to think about the intelligence of evolvability. And an example of something I'm just putting up here is how quickly we evolved from being pretty small brains to pretty big brains over a very short time. Is this anything to do with it? I don't know. But this is an example of the kind of thing that may be due to the fact that we've got very intelligent sort of evolvability networks in our bodies. So anyway, thank you.